Thank you so much. And then I would like to ask any question and then from the audience. Okay, and please, yeah.
Good afternoon, and nice presentation. And I have a questions to ask Robert Knight. And you are the expert in the bioinformatics, and also you mentioned that the AI in the use in the analyze in the microbiome. So I want to know because you mentioned that, I want to know whether you have some suggestion, excluding the AI in the regression or the classification model, excluding these two directions, whether have the other, some the treatment or the simulations in the gut microbiota and the disease, because I am focused on the CRC or the other digestive disease analyze. Thank you.
Great. Thank you for your question. So yes, we are indeed using AI to improve on classification and regression over random forests. So there is a recent paper on TRPCA that we just put out, which is transformer-based improvement on the RPCA method for analyzing compositional microbiome data. In addition to that, we are using other attention models where we can build a foundation model of how collections of ribosomal RNA are structured in different microbial communities. And we think the advantage of the foundation models, both at the 16S level and at the whole genome level, which we are working on now, will allow us to not only improve classification and regression, but also to do some of those generative AI tasks that I mentioned. I think so. In particular, being able to do things like arithmetic in the embedding space. So in the same way that you can do semantic arithmetic on words and concepts in an embedding space, we think you will be able to do that kind of thing with microbiomes. So, for example, to project what would your current microbiome look like if you had fatty liver disease or IBD, or could you change your microbiome with diet in a particular way? And as a separate step, could you use AI to build the model of what that diet should look like? So we think there are many different applications.
Okay, thank you.
So thank you. So one question for Dr. Yao. So from your data, I mean, it seems to me naively that clinical trials should be very readily designable for that, right? You just have two groups of cancer patients match somehow, and then one group has some long-term antibiotic, and then you see the prognosis, basically. Is that sort of work being done?
Yeah, thank you for your questions. And I think, yeah, it's very essential to, in the clinical trial, to test the antibiotics to deplete intracellular bacteria or extracellular bacterial function and see the outcome, the treatment outcomes. And we're trying to do that, but it's very hard to get enough human cancer patient recorded. And we hope that in the near future that we can figure out this. Thank you.
Thank you. I have a question to Herbert. Okay, I think it's interesting to identify two phenotypes of MASH. Do you think the gut microbiome tablets may contribute to phenotype of MASH?
Well, I'm convinced it contributes, but of course the challenge is we have interaction with dietary components, and are dietary components the major drivers, or is it microbe-based or metabolites released by microbes? And both different worlds might, for example, activate the same mechanisms in innate immunity, let's say targeting NLRP3 or certain cytokines. So it's challenging and to find out, because it's so highly interacting, dietary with microbes, microbes with diet. So I think that's one of the major challenges we're facing now in understanding the disease.
Yeah. Okay, please. Yeah.
Good afternoon, and thank you to all the professors for a very insightful presentation. My question is for Professor Herbert. So in your talk, you mentioned that the liver inflammation and also muscle is also associated with the gut microbiome. So I just wonder, there are a few probiotic cocktails that are under clinical trials right now. So may I ask, what's your perspective in gut modulation? Like, if we modulate the gut microbiome, maybe it's a good strategy for maybe in personalized medicine as well, because the gut microbiome also have varies between populations, and I wonder what's your opinion on that?
Thank you very much for this very important question. I have to say, preclinically, we have so many wonderful data when we use probiotic 1 to X and Z, and it works preclinically. But in a clinical setting, it's much more challenging. First of all, we need studies at least over 1 year. Second, we need sponsors. So industry so far had not been motivated to finance this type of trial. And third, my personal belief is that maybe we should better understand what is going on and then probably target metabolites, because maybe targeting or using 1 or 2 bacteria might not be sufficient. I would rather go for metabolites. And there is a nice example recently demonstrated. There is a metabolite with the name imidazole propionate. It's histidine derived, and it's generated by certain bacteria. But imidazole propionate is very pro-inflammatory. There was a nice Nature paper now showing that it plays a major role in atherosclerosis, and I told you atherosclerosis is the #1 cause of death in muscle patients. So this could be, for example, a very attractive target. And I think we should go for such targets. And in muscle, I have to say there is still a lot to do, because we have not yet identified many relevant metabolites derived from the microbes. Thank you.
Thank you.
Okay, and please, next one. Oh, so many. Okay, and one by one, please. Yeah. Yes.
I also have a quick question for Professor Knight. So first, thank you for a very comprehensive talk. So my question is that you have mentioned a very interesting slide about that the lean and obese patients are more determined by the microbial genes than the human genes. So as we know, the tumor always occur with numerous genetic mutations and also the microbial dysbiosis. So I want to ask, in your opinion, which one played the dominant role in the tumor development, the genetic mutation or the microbial dysbiosis? Thank you.
Yeah, great question. So for the lean versus obese question, it is not just us. Many other people, including Iran Segal's group, have done direct comparison of the human versus microbial contribution to that phenotype. And in obesity, it is very clear the classifier accuracy is much better, and you need far fewer people to see it if you are looking at the microbial genes than the human ones. For cancer, I don't think we really have the data yet to do the same kind of contribution about microbial versus human genetic causes. And additionally, there is the issue about whether you're looking primarily at the gut microbiome and metabolites and immune cells that are produced or trained by the gut microbiome that then circulate through the rest of the body, or whether you are looking at microbes that literally translocate inside tumors, as you heard in the last talk. So I think it is a very interesting question, but not one where we have the data to decide yet, although many people are working on it, and if you're looking for projects, it would be a fascinating one to tackle.
Okay, thank you. Next one. Okay. The boy first, and then the girl in the front.
Thank you. My question is for Professor Rob Knight. So your talk is very interesting, and I was very interested about the slide the microbial changes from the same patient, same person who undergo different healthy stages. So my question is that which bacteria are responsible for that change, and whether these key bacteria are the cause for the change, for the transition of the microbial, the healthy state, and or are they the result of this healthy status?
Yeah, great question. So I would have to go back and look at the data to tell you the individual names of bacteria that were strongly associated with those different states. I don't remember off the top of my head, but what you are seeing during the changes at different times from that one person are almost complete turnover of the microbial community. So there is essentially nothing the same, especially in his post-surgery setup. His post-surgery state is almost completely non-overlapping from his pre-surgery state. And the other thing to note is that we don't know causality in that time series. So typically, what we know at the moment is we know what bacteria are correlated with the state changes, but we don't know which of those are driving the physiological changes. And I'm not sure whether we're going to be able to get that out of one person's data. We're collaborating with George Sugihara at the Scripps Institution of Oceanography, who has done a lot of exciting work on inferring things like ecosystem collapse parameters from time series data. And the reason why we can apply that now, which we could not do in the past, is that we are now using reference standards for the DNA. So we get absolute quantification in terms of copies per gram of the different kinds of bacteria instead of the relative abundance data that are common in the microbiome field. So we are starting to do that, but we don't have results yet. The other possibility is using things like Granger causality over the whole time series. And even with hundreds of time points, we do not have quite enough data to do those kinds of causal inferences robustly. Typically, those have been more successful in data sets with thousands to tens of thousands of time points. So we have some fundamental limitations. What we really need is more people to be willing to collect their microbiome samples every single week, like Larry has been doing, or ideally every single day. And a lot of what we are trying to do at the moment is to make that cheap and feasible enough that you could actually do it. So for example, for my own time series that's been running for 15 years now, we recently switched to these pre-barcoded tubes. So this one has not been used yet, but essentially what you can do, they have a little barcode on the bottom, so you can scan racks of 96 of them at a time, and the whole DNA extraction process and process of getting them from this format into 96 well and then 384 well plates is automated. So that kind of thing dramatically reduces the cost of the rest of the project. Like I showed you, the cost of sequencing has already been dramatically reduced. And so we are trying to figure out how we can make it feasible to do this sort of thing at scale in a way that individuals could reasonably do it without a lot of difficulty in infrastructure. Now, it is pretty difficult to get people to collect their stool even once in a clinical setting, as you heard in the other talks, and getting them to do it every day is even more challenging. Getting a blood drawer every day, I think there would also be a lot of difficulty getting people to do that. And so trying to find what biospecimens you can collect and analyze in a reasonable way for these kinds of studies is also an ongoing topic of interest in our lab and in my center.
Thank you so much, Rob. And due to the time limitation, we have to close this Q&A session. May I invite all the speakers to stay in the stage and have a group photo and with Professor Denslow, the chairman of Future Science Prize Week.